Welcome, everyone. Today, we will explore a new integrated theory of evolution, combining insights from polygenic adaptation, epigenetics, and the speculative but fascinating concept of morphic fields. By the end of this lecture, we'll understand how these processes can work together to explain complex and rapid evolutionary changes in organisms, using the evolution of the human esophagus and larynx as our primary case study. Let's start by outlining the foundational concepts we'll be working with.
Polygenic adaptation, the genetic engine of evolution.
We begin with polygenic adaptation, a well-established concept in evolutionary biology. Polygenic adaptation refers to the idea that certain traits, particularly complex ones like height, intelligence, or the structure of the vocal tract, are influenced by many genes working together. Instead of a single master gene driving the development of these traits, thousands of small, subtle genetic variations combine to shape them. Natural selection acts on these variations across generations, slowly pushing the population towards more advantageous versions of the trait. In humans, for example, we can imagine how tiny shifts in hundreds of genes might make our larynx a bit more flexible, or our vocal cords more capable of producing varied sounds. In our case study, the evolution of the human larynx and esophagus, this gradual adaptation could explain how, over time, our ancestors became better at vocalizing. As social communication became critical for survival, polygenic selection might have favored individuals who had slightly better control over their voice, allowing for richer and more complex forms of communication. However, polygenic adaptation typically occurs over long periods. Yet, in the case of human speech evolution, changes happened quite rapidly. This is where our next concept comes in.
Epigenetics, the environment's immediate hand on evolution.
To explain faster evolutionary changes, we turn to epigenetics. Unlike genetic evolution, which alters the DNA sequence itself, epigenetics refers to changes in how genes are expressed. These changes are often triggered by environmental factors, everything from diet to stress to behavior, and they can be inherited for several generations, though they don't alter the underlying DNA code. Think of epigenetic modifications as tiny molecular switches that decide whether certain genes are turned on or off. For example, imagine early humans who began to rely more and more on vocal communication for social interaction and survival. The use of vocalization could have activated certain genes related to vocal cord and laryngeal development, modifying how these structures grew within an individual's lifetime. These epigenetic changes could then be passed on to their offspring, giving future generations a slight edge in vocal ability, without needing full-scale genetic mutations. This helps explain why the larynx and esophagus evolved so quickly in response to the pressures of speech. Epigenetic mechanisms could have worked in parallel with genetic evolution, allowing our ancestors to adapt more dynamically to their environment.
Morphic fields, a new layer of biological organization.
Now we introduce a more speculative but intriguing concept, morphic fields, proposed by biologist Rupert Sheldrake. While controversial, this idea provides a fascinating way to think about biological patterns and forms. According to Sheldrake, morphic fields are non-physical fields that act as blueprints for the development and behavior of organisms. These fields carry a kind of collective memory, influencing how similar forms or behaviors are expressed across generations and species. In our case study, a morphic field for human vocal structures could help explain the rapid and coherent evolution of the larynx and esophagus. If morphic fields exist, they would act like organizing templates for these body parts, guiding their development toward a form that's optimized for speech. Here's where this concept becomes powerful. Rather than evolving randomly, our laryngeal and esophageal structures might have been guided by a pre-existing organizational field that knew how these structures needed to evolve for advanced vocalization. This would allow directed, coherent evolution to occur much more quickly than if random mutations and selection pressures alone were responsible.
Integrating the concepts the Triadic model.
Now that we've introduced polygenic adaptation, epigenetics, and morphic fields, let's see how they integrate into a cohesive model of evolution, one that can explain both slow, gradual change and rapid, directed adaptation. Step 1. Polygenic adaptation as the genetic foundation. In our model, polygenic adaptation provides the genetic baseline for evolution. Multiple genes across the human genome contribute to the development of the larynx and esophagus, traits critical for speech. Over generations, small shifts in these genes, favored by natural selection, would gradually refine the vocal structures. Step 2. Epigenetics as a rapid responsive mechanism. As our ancestors began relying on speech for survival, epigenetic mechanisms acted as a more immediate response system. The environmental demands of social interaction, increased vocalization and communication, triggered epigenetic changes that could modify gene expression in real time. These epigenetic modifications, passed down across a few generations, accelerated the development of speech-related anatomy. In essence, while polygenic adaptation was doing its slow genetic work in the background, epigenetic changes provided a faster, more flexible layer of adaptation that could respond to immediate environmental needs. Step 3. Morphic fields as an organizing blueprint. The most novel element of our model is the role of morphic fields. While polygenic and epigenetic mechanisms work on the genetic and molecular level, morphic fields add an organizing force that shapes how these processes unfold. Think of morphic fields as a template that guides how the larynx and esophagus develop, in response to both genetic and epigenetic influences. This template, or field, could contain a collective memory of how vocal structures need to evolve for speech, based on the experiences and behaviors of previous generations. By integrating morphic fields into our model, we suggest that evolution isn't just a blind, random process. Instead, there may be a guiding pattern or form that organisms tap into, accelerating and shaping the way they evolve.
Case study: human laryngeal and esophageal evolution.
Let's return to our case study to see how this integrated model explains the evolution of the human larynx and esophagus. Polygenic adaptation gave our ancestors a genetic framework for vocalization. Over many generations, selection acted on small variations in genes that control the muscles, tissues, and nerve systems of the larynx and esophagus. This slow genetic change laid the foundation for more effective communication. Epigenetic changes accelerated this process. Early humans who relied heavily on vocal interaction triggered epigenetic changes that modified how their vocal anatomy developed. These changes could be inherited across generations, allowing the vocal structures to adapt quickly to the growing demands of speech. Finally, morphic fields provided a blueprint for the rapid, coherent evolution of these traits. As more humans developed speech, the morphic field for vocal structures strengthened, guiding the evolution of these structures in a more directed manner. This explains why the evolution of the human larynx and esophagus appears to have occurred faster and more coherently than would be expected by random genetic mutation alone.
An integrated theory for evolution.
To summarize, our integrated model of evolution combines polygenic adaptation, epigenetics, and morphic fields to explain both gradual and rapid evolutionary changes. Polygenic adaptation provides the genetic foundation, a slow, cumulative process shaped by natural selection. Epigenetics acts as an immediate response mechanism, enabling organisms to adapt quickly to environmental pressures by modifying gene expression. Morphic fields add an organizing force, potentially shaping evolution in a more directed way by providing a collective memory of form and behavior. This triadic model gives us a richer understanding of evolution, moving beyond a purely genetic view to include environmental feedback and potentially non-physical fields that guide biological development.